what I'm going to talk about today is, and I should also note, if you actually read abstracts, I said in there I was going to talk about arborescent lycopsids on top of everything else, and that's too much. That's too ambitious. So no arborescent lycopsids, but you just got some of that anyway. So what I am going to talk about is just a general issue of, we can look at a forest like that. We can think about Paleozoic plants. And it feels kind of right. It's a dragonfly, it's not a bird, but it feels like a forest that we can kind of recognize. And is that okay or not, that comfortableness we have with this? And I'd argue in a lot of cases it really isn't. Because when we think about how these systems function, we have to think about the modern world. And the modern world has been utterly overrun with flowering plants. Right? There's nowhere you can go on the Earth's surface where it's not 90% of the species are not flowering plants. You go to a coniferous forest, most of the species are still going to be angiosperm herbs in there. And is that okay or not? Is it okay to look at a modern angiosperm system and then think about that, applying that back through time? And I'd argue it's not. And that actually really matters. And I know this is a plant meeting, so I don't have to say this, but I think it's important to just say it. Animals are cute, and that's fine, but plants are the carbon cycle. They're all of the biomass, they are the water cycle, they are climate, they are the atmosphere. Right? So the evolution of plants is the evolution of the Earth's surface, and that matters. And that's very much tied to the evolution of the plants involved, and a lot's happened over the Phanerozoic. And ultimately, the point is to talk about back here, that yellow circle, back in the Paleozoic and the early plants. That's the whole point of this symposium on some level, although people are going earlier than that still. But to get there, I have to first ask, what about those guys in green there, the flowering plants, that now have completely dominated the world? And the evolution and the turnover of all these different groups, it really does matter for the climate cycle and for the Earth system. I almost took out the next couple of slides because we've seen some climate models and Earth system models at this point. But I think it was still important to include these, as I'll explain. So what this is showing is the impact of flowering plant evolution on transpiration. They transpire water, is expected about 4 times more than any other group of plants. And that has real impacts upon climate and precipitation. This is showing tropical South America, and at the center of that bullseye, what this is a difference map. Everything's the same between the modern world, modern biomass, modern sea surface temperatures. The only change is transpiration. And there's a loss of up to 1,500 millimeters of rainfall and an 80% reduction in rainforest area in tropical South America just by changing the plants. And it's important to include this, even though we've seen some of this stuff before, is that the efforts we just saw with Isabel and in the Ordovician yesterday is much more heroic because we're talking about a long time ago. With different plants, but also different geography, different atmosphere, different sun. Whereas this is only changing the plants, and that has that large of an impact. We've more recently been generalizing that. This is a paper now in review where we did the exact same thing as in that previous paper, but we're stepping it down even further, where that top panel there is an equivalent panel, but it's showing you globally. The loss when you have modern vegetative biomass, but step down transpiration to vascular plants other than the flowering plants. This bottom panel closer to me is albedo is the same. The plants are still there, but they're not allowed to transpire, and the effects are larger still. That's kind of what would happen if you took out the plants that were there in the Carboniferous kind of thing. And then the last one is if you just remove them entirely, so that the albedo effect, showing that even just the albedo of plants is a huge impact. And the stripes, the red and blue stripes reflects that the location of the tropical rainy belt, the intertropical convergence zone, is being shifted up and down. That's why there's also more rainfall in some areas, because that's being shifted. Okay, so that was based upon transpiration levels in flowering plants being higher. Why do I say that? You started to get a little bit of that introduction to this in Isabel's talk, and I'll now go into it in more detail. Basically, it comes down to the fact that flowering plants have a lot more veins than other things do. So in this here, the leaf on the top is a fern, and then the leaf underneath is a glorious angiosperm with all those crazy veins in there. And you can't even see the finest veins, probably. There's a lot going on there, whereas you're seeing everything in the fern. So why does that matter, right? So what the plant is doing photosynthetically is it's taking in carbon dioxide through the stomata, and it cannot help but lose water in the opposite direction when that happens. So the plant has to be able to replace the water it's losing, or it has to shut the stomata. It can't just allow itself to desiccate like that. And so the hydraulic capacity of the plant is going to greatly influence the photosynthetic capacity of the plant. And it turns out that a large fraction of the resistance for the entire plant is just in the last little centimeter or two represented by that leaf. It's at least a third. It can be up to 80% in some cases. So the water comes in from the roots, up the stem, out the branch to the leaf. It's still largely concentrated in the leaf. And the reason for that is that the leaf is the one place where it's leaving pipes designed for water transport and going through a tissue that has a completely different function, which is the photosynthetic tissue. So that's a resistance bottleneck for the whole system. And anything you can do to lower that overall resistance is going to allow greater photosynthetic capacity. And the easiest way to lower that resistance is to decrease the path length. This is a vein cross-section here, a leaf cross-section. So the orange there is where the vein is. And if you can shorten that path length from the vein to the stomata, that will increase the overall capacity of the plant. And the easiest way to do that is to increase the vein density. You have more veins, those path lengths are going to be shorter. So this originally kind of came from some work by Tim Broderick and colleagues. And so what they showed is that if you shorten that path length, so that's vein stomata path length on the x-axis there in the first thing, that will increase leaf conductance. And if you increase leaf conductance, that will increase photosynthetic rate. Right? And so that's assimilation, photosynthesis. That's true of transpiration as well. Here, the x-axis is flipped relative to what you just saw, but we just saw distance from vein to stomata, whereas this is vein, the total density of vein, so kind of how many millimeters of vein per square millimeter of leaf surface. This is kind of the inverse of that, and that's showing a good relationship with transpiration. More veins, shorter path length, more transpiration, more assimilation. And the key here is that that's something we can actually measure in the fossil record. And so that's very kind of important because, as I've said, the world's been completely nuked with flowering plants. And it seems fair and reasonable to suggest that a group like the cycads, which aren't doing very much photosynthetically now, maybe they were doing more when they weren't marginalized by all those flowering plants. But we can look at their leaves in the fossils and say they're the same. There's no difference here. And that's a general truth for the lineages that we have with us now. Their fossils kind of look the same. They're not doing anything radically different. So cycads are slow growing now. They were slow growing in the Triassic. And I see no reason to think otherwise in that regard. So that's an important conclusion right there, that we shouldn't leap to hoping to get more out of these organisms in the past without knowing more. And then because we can measure these things in the fossils, we can just look across all plants through all of time and say that flowering plants really are doing something distinct from anything else ever has. So don't worry about the colors. That has something to do with leaf development and architecture of the venation system. But looking at the overall kind of distributions of these levels, the blue groups at the bottom, those are the flowering plants there. And if you look at the graph, that inset graph there, what that's showing is vein density through time, and it's very stable and very low. The average is about 2 millimeters of vein per square millimeter of leaf surface. It'll go up to 4 or 5. It'll go down to 1. But that's it, and it's very flat until flowering plants come along and they increase the average, the mean, to 8 or 10, and they can go up above 20. So both the mean and the maximum is increasing by a factor of 4. And that should really matter for these things that I've just discussed. There's one kind of important caveat to say there is that the leaves aren't magic. If you take one of these leaves and you staple it to a cycad, you're not going to get a magic cycad that's going to grow really fast. You're going to get a dead leaf because it's integrating the characteristics of the whole plant, the vessels that angiosperms have and all these other characteristics, difference in stomatal behavior and everything else is going on that's different. This is integrating that and providing kind of an indication of all these other things that have changed along with it. Okay, that's kind of an important kind of caveat to say. But angiosperms are quite distinct from everybody else, and that should affect both transpiration and assimilation, which should have large impacts. I showed you the transpiration in the rainforest diagrams. What I'm going to focus on is the productivity side of that, because that's a harder thing to discuss, because other things are going to matter for productivity, like carbon dioxide levels. When we did the first papers on that, it was not immediately as obvious how to handle that. But two of my colleagues on that original work, Tim Broderick and Taylor Field, had a follow-up paper of their own where they looked at productivity. And they tied it to photosynthetic models that have existed for a long time. And they got vein density in with those. They tied vein density in with that. And they came up with an interesting hypothesis that, that the way flowering plants became what they are was tied to a drop in carbon dioxide levels. So that at high carbon dioxide, there's no reason to have high vein density because it doesn't do you any good. That's this top curve over here. And it's only as CO2 comes down that there starts to be a point for this high vein density. That was their idea. It bothered me when I saw this paper. I loved all the measurements they did, and I loved that they had integrated it with the photosynthesis. But the problem I had with it was the other diagram there. Of the basis for all that is saying that if you have enough carbon dioxide in the system, any fern will be doing as much as a sunflower is now. And that's the part that just felt wrong to me, right? It's hard to imagine that ferns really are ever going to follow that upper path. And that kind of feeds into an issue out there, right? There's a community that has put a lot of effort into studying carbon dioxide and how that's impacted plant evolution. And they communicate very well and they're very productive. And there's kind of scattered other papers out there that really isn't consistent with that. But you know, this is more kind of disjunct and no one's really paying attention. For example, like I know in my life, a very influential paper is that top paper by Bond, The Tortoise and the Hare, talking about angiosperm ecology versus gymnosperm and conifer ecology. I love that paper, but if you think about it, the arguments that he presents there wouldn't really work in a world where productivity was much higher at times of high CO2, and that the differences between lineages were kind of flattened out, right? And then, you know, the only other work I did when we first did this, I wasn't really thinking in these terms, but you know, I was implicitly disagreeing with it too, even though I hadn't really thought it through one way or the other. So I thought it was interesting to have a chance to address this discrepancy, right? And have a situation where rather than, you know, using fossil plants to either assess carbon dioxide in the past or use past carbon dioxide to, you know, for a viewpoint on how plants evolved, have a chance to actually then return it to the fossil record and ask, well, does that work? Does that actually jive with what the record looks like? And the Broderick and Field paper did a very good job of providing that kind of an opportunity. So what they did is that they, as I just explained, they were very influenced by this plateau here, saying that high vein density isn't helpful at high CO2 concentrations. What I found more interesting was not the plateau, but ramping up to that plateau, because that's the range that any plant can occupy. Ferns do it, cycads do it, anybody does it. And within that range, the bottom half of that range that any plant can do is going to be at a huge disadvantage if this works. Just a small incremental increase in vein density, you know, makes you from a modern fern to a modern sunflower. And that suggests that at times of high carbon dioxide through time, if this model is correct, the bottom half of that vein density range should be evacuated. So that the lower bound should be increasing and decreasing through time. Because you cannot afford to miss out when everything else is on top of that plateau there. So we can look at vein density through time, and we don't see that at all. So this is Berner's geocarb sulf output. And for various CO2 concentrations, there's the distributions of vein densities at different points in time, and it doesn't matter. There's no pattern there. So at first, I was happy and I felt done. that didn't work out, and I didn't think it should work out, and it didn't work out, and that's good. But we got to thinking that, you know, well, why? Why didn't that work out? There was no obvious thing wrong with what they had done, and the photosynthetic models involved are the things that people have been using for decades very successfully. So what's the problem here? Actually, we should really think about what's going on, and is there a way to reconcile what they had done and what we had done with the actual fossil record? So we went to the model they used, which is kind of based off of Farquhar's standard model. And what they did is they defined leaf conductance in terms of the vein density, and then they looked at the flow into the leaf as being proportional to that conductance and the water potential gradient, and then the flow out of the leaf, the evaporation, transpiration, is going to be proportional to the vapor pressure and the stomatal conductance. Flow in has to equal flow out. So that allows getting stomatal conductance in terms of vein density, and then they can plug that into a model of photosynthesis based on physical constraints. There's an alternative model based on biochemical constraints. You solve those simultaneously, and that's how you get those curves. Okay, so what went wrong? And so we try to look at, are there things that we could correct and how this was approached? And the first thing we tried to do was in their model, originally, they had leaf thickness was fixed. All leaves were the same thickness. And so what we did is we allowed that to alter freely to the vein density. And that's for reasons that I'll explain in a little bit. And that was very gratifying. So they had actual empirical data, which are the dots, and the solid line is their model, and our line kind of works better with it, so we were happy with that. But that correction is helpful, but it doesn't change these graphs at all. There's still a huge disadvantage to having a low vein density under those circumstances. Then we tried varying other things. They didn't play with oxygen at all, and there was a typo in how they'd done oxygen. And so we tried low oxygen and so on. That didn't make a difference. We allowed stomatal conductivity to also be reduced following Franks and Bierling at the higher CO2 concentrations as well. And none of that made any difference. We also, just for the hell of it, tried a more continual range of carbon dioxide concentrations, up to 2,500 ppm. And it just shows that once you get elevated, there's this incredibly steep gradient. And there's, regardless of how high it gets, you shouldn't be in the lower half of that range. So none of this made any difference. None of this helped. There was one thing, though, that does provide a solution, which is maybe a fern can't be a sunflower. If we just lower the maximum carboxylation rate possible, that gives you a different series of curves. And if you lower it enough, what happens is you still get a plateau. But that plateau now encompasses the entire range of vein densities these things can do. So vein density is no longer relevant to assimilation rates under these circumstances. It's still going to matter for transpiration depending on where you are and that kind of stuff. But all that requires is just saying that maybe all these lineages aren't capable of that very high level of photosynthesis that was suggested. And that's actually consistent with measurements in modern plants. Yes, there can be very high fertilization and high rates of carboxylation among angiosperms. But if you look at various non-angiosperms, the ones with these low vein densities, they don't elevate that much, right? So that's actually consistent with living plants. It's also actually consistent with the fossil record in that there is one lineage that did show the pattern I was looking for, and that's the flowering plants. So they're the white dots here. And as they evolve over the Cretaceous, as they evolve some of these traits, the high vein density and so on, they go from completely overlapping with ferns to evacuating that lower half of the zone as we had originally hypothesized they should do. So if anyone's responding to carbon dioxide correctly, it is the flowering plants. It is not the other lineages that just kind of don't change through that. And so kind of one last thing to say about that is that it's maximum carboxylation in the model. It doesn't have to be maximum. It doesn't have to be that. It doesn't have to be rubisco. It could be any number of things. It can be photoreceptors. It can be the rest of the Calvin cycle other than rubisco. It could be nitrogen and phosphorus. Nitrogen came up yesterday in a question as far as high assimilation rates. There's lots of things that could limit the maximum ability to do photosynthesis. And this is just kind of subsuming all those under one variable. But something's doing it, limiting all these other lineages. Even leaf anatomy is relevant, as I'd like to show quickly now. So this is some work that was done previously. I was not involved with this, but it was done by a close colleague of me, Maciej Wieniawski and his colleagues. They made artificial leaves out of gel, and they carved little channels for water to evaporate from the thing. And what they found is that the more channels you have, the more water you evaporate, but it's also proportional to the thickness, so that you end up plateauing. And what that shows is that there's an optimal kind of distance between the veins and the stomatal surface where the evaporation is occurring. They should be about the same, these two distances here. And if you have too many veins, then it's not going to do you any good. I'm looking at the wrong thing. I'm too close to the screen. So you can have a thick leaf with a low vein density. You can have a thin leaf with a high vein density, or you can have a thick leaf with a high vein density if those veins are offset towards the stomata. But you should not have a situation where you have a thin leaf and very widely spaced veins because you're going to dry out in the space in between. And you shouldn't have a lot of veins far away from that surface because that's just wasteful and dumb. There's no reason to do that. And then they looked at a whole bunch of plants, and everybody fell on this wonderful one-to-one line, which is exactly where they wanted it to be, which was cool. The problem with that is that this was when we were also measuring vein density in a whole bunch of ferns. And well, where are the ferns, right? The ferns belong right there. They should not be on that line properly. And I even had a kind of hypothesis about what went wrong here, was that I figured that they had grabbed a bunch of stuff from exposed environments, like on university campuses and stuff like that, where it's important to be on that line. But maybe ferns are largely in less exposed environments where it's okay to fall off that line. So you end up with a situation like this. You should still never be over-invested, the thing with the X. And if you're in an exposed environment, you have to be on the line, but if you're in a sheltered environment, you can fall off the line. And we measured a whole bunch more stuff, and it's not a line, it's a triangle. And first, it is gloriously how perfectly they did the math, because the bottom half of this is still not occupied at all. So they really did get the understanding right of where the line should be and so on like that. But a lot of things do fall off that line. And it turned out that there was a very interesting phylogenetic signal to it all, which is, yeah, the ferns are where they should be. And then gymnosperms have thicker, they still have low vein density, but they have much thicker leaves. So by virtue of thicker leaves, they can end up on this line with thick leaves. And also, you can barely see this, but the dots with the orange around them are fossils. And all the fossils we looked at are completely consistent with the living versions of the same groups. So there's kind of some consistent physiology going on here. The big surprise was that all angiosperms just fell right on the line. Didn't matter. Didn't matter where they were, if they were in the shade or not, they were on that line, except for basal angiosperms. They look like ferns. They're the red dots. So early in angiosperm evolution, this is prior to high vein densities and everything else, when they are kind of things living in the shadows in tropical forests. They were kind of like the ferns. But as they got these more derived traits, they all end up on this line, whether in this exposed environment or not. And so you can kind of see this here. This is taking the same information, but looking at the ratio. So if you were on the one-to-one line, you're now on that red line there, that thin red line. And so this allows us to show it phylogenetically. And we can see non-angiosperms are all well off that line. I mean, there's a range, right? So if you get an exposed tree fern, it will be closer to that line than any other fern will be. So there's still a range there, and closer is still going to be better. But the only things that are consistently there are derived angiosperms, right? So it's not Nida, not Piperales, but the Laurales, right? So it's within the magnoliids, it's within derived monocots, it's within the eudicots, and not more basal lineages there. So we set up this kind of very interesting alternative ecologies that say that things have kind of been consistent in a lot of ways, where ferns, in theory, are capable of high gas exchange because they have thin leaves, but they're in the shade where there's not really an opportunity to do that under most circumstances. A cycad, full sun, but these are thick leaves. So they are not limited in sun, but they are limited in their gas exchange potential because of the thickness of those leaves. What flowering plants did is this really weird trick of they took leaves that belonged at the bottom of the canopy and figured out how to put them on the top and maintain them physiologically. Right? So they really are doing something weird and different. So when we look at these kind of graphs of productivity through time that are out there, I think the top half of these graphs didn't really happen. Now, given what Isabel just talked about, we're not in disagreement, right? So where she had things, when she was talking about 10, right, there's 10, right? I'm not, that's, when I say high productivity, it's not the same thing that Isabel means when she says high productivity. But these very elevated levels, hypothesized for periods in the past, I don't think those were really an option. And that even feeds into things like silicate weathering, where if you look at Berner's model, he's done sensitivity for these things. And he's demonstrated that in his model, if you have too much CO2 fertilization, you don't get a CO2 curve anyway. It just flattens out. So there can't be that much productivity in that regard. Okay, the Paleozoic, where I was intending to go with this in the first place. So what does this say about when then looking back to these early ecosystems and whether we can use modern ecology? Well, let's look at a graph from modern ecology, right? There's a graph from modern ecology if you know your modern plant ecology. There's Grimes' triangle, right? And you have the stress tolerators, you have the weeds, and you have the stately competitors. And then this is defined by their ability to handle disturbance and competition and stress. Two of those three vertices are based upon high productivity. And I would argue that that is an angiosperm trait in that regard. And everything is going to be compressed down towards what we now consider to be stress tolerators. Right? Note, this is still a triangle. It's not a dot. Right? So there's still variance there. And Isabel's medulosans and stuff like that, I agree. They have the highest vein density of any of these things. They would be kind of on this line here, that farther line there, but it's still very much down from what we would consider from modern systems. And this has some kind of funky implications, right? So we're kind of talking about a world without weeds. Right? What are the non-flowering annual plants? There aren't any, right? There's plenty of herbaceous plants, but ferns are typically going to be perennial. There are exceptions. And among seed plants, it's just a flowering plant thing. In the fossil record and living things, there are no annuals outside the flowering plants, which is, I think, a reflection of this. Whereas if productivity really was fluctuating enormously with CO2 through time, you'd have a different, I think there would be a different pattern resulting for something like that. And we can also kind of look at this in more of a kind of a time slice kind of way. This is the Westphalian B, so this is less than 1 million years. And I'm only looking at you people here. This is the Yorkshire coal basin and stuff like that. So this is only England. And I just sampled the heck out of it, right? So this is as well sampled as any similar geography in the modern world, probably better actually. And this is the range we see. The colored bands show us the range from the flowering plants now. And everything is, the best guys are still what would be considered an obligate shade plant among flowering plants, right? That is how depressed everything is in that regard. There's still range, right? And yeah, the Megillosins are higher than everybody else, but that's still very much in what is a shade plant among modern flowering plants. And that has some weird implications, right? So yes, the range of ecology is going to be much, much kind of contracted in that regard. And that might have some relevance to how ecosystems were functioning back then. There's also some other kind of fun things I think to think about, where if every plant looks like a shade plant, how do we think about the evolution of plants in the first place, right? The classic notion for all of this is always that plants are reaching for the light and overtopping each other and shading each other and competing for something. I think all of these plants are getting more light than they can handle. They don't know what to do with it. They can't use all that light. They're not competing for light. And that's kind of this, that trope is classic, right? I mean, so that's, when people think about the evolution of plants, in the back of our minds is always this, the modern world and how you have a dune or something, and then you have some annuals come in, and then more herbs, and then eventually you get this trees and the pioneer trees, and then you get the climax forest, right? That's all about competition for light. And I mean, just to say that I'm not immune to this, like this is from my thesis defense, right? I've used this slide 1 million times, and I'm clearly thinking about that when I put that in there. But I think that's very misleading. I do not think these things were competing for light. If that's the case, then why did trees evolve, right? What's the point of a tree if it's not going for light? There's different potential answers to that. If Carl Nicholas was in the room, he would say it's for propagule of dispersal, which is fine. I don't think that's irrelevant. I don't think it's wrong. I don't think it's the whole story. There's potentially advantages just to being larger and to having more leaves and more sporangia and all that kind of stuff. What I think is the most compelling option is that it's not really about growing up, it's about growing down. That this is really about the evolution of deep rooting. If you want to have deep rooting, you have to live for a long time in one spot. And if you're a plant, you cannot help but grow. And if you want to grow in one spot, you got to grow up if you want to grow down. So I think the impetus for the evolution of trees might be as much about what's going on in the substrate as competing for light above ground. And we can see that in the modern world where this is looking at rooting depth in different ecosystems. And there's very deep-rooting plants there, right? And some of those deep-rooting plants are herbaceous grasses and stuff, but they're grasses, right? That is angiosperm productivity there. In the absence of elevated productivity, if you want to be deep-rooting, you basically have to be a tree or shrub living in one spot for a long time. And so I think that really could be shaping what's going on there, that it's about root evolution and deep-rooting. It is not about competing for light. Okay, so there's kind of a summary of what I was saying, that I think we can't just think about plants as plants and as a generality. We have to really think about what plants are involved and what they could have actually been doing physiologically. And we have to be very wary of, vigilant about what's creeping in from the modern world and the way we're thinking about it without necessarily thinking about it. And I think we can think about these things in real terms because there are ways to get at physiology from the fossils. Isabel talked about a variety of those, and this is another option among those things. And I think that productivity would have been lower. It never was, I don't think it was, early in the history of angiosperms, early in the Cenozoic, yes, CO2, productivity could have been higher than it is now, yes. But I don't think there's any point prior to angiosperm evolution that it was higher than it is now. It was lower, it was not higher. That kind of consistently low productivity relative to what angiosperms do now, I think is relevant to a lot of these kind of very basic questions about how plants evolved in the first place. Okay, I shall stop. Thank you.